Hello everyone, and welcome to episode 97. Today's show marks a new beginning of sorts, for several reasons. First of all, we're done talking about animal taxonomy. In the previous series, we explored the impressive biodiversity of the protostomes and the deuterostomes by working our way through all of their internal phylogenies, their family trees. In theory, it was pretty interesting stuff, as it elucidated a lot of the evolutionary relationships between groups of mollusks, or arachnids, or amphibians, or reptiles, or what have you. As far as we're concerned for our biological narrative here, we've established the technical context for pretty much every group in the animal kingdom. This takes us to the second reason this episode is like a new beginning. Now that we're done with taxonomy, we're still going to be looking at the animals, but this time we're going to focus on their anatomy and physiology, their development and life cycle, their behavior and their ecology. In other words, we're going to look at how these animals exist in the world and survive and interact with their environment. These are going to be some super exciting episodes that really dig into the nitty-gritty details of the animal's life, including their birth and growth, their adaptations and the selective pressures working against them, and how they kill their prey, or get killed by predators. And where the taxonomy episodes were rather large in scope, like with a single episode to cover all the crustaceans, or a single episode to cover all the fish, or all the mammals, these new episodes will be much more specific. We'll have an entire series on the crustaceans, and another series on the fish, and on the mammals. Each episode will examine a group within the clade. So for example, the crustacean series will have an episode on lobsters, and another on crabs. The fish series will have an episode on the sharks and rays. The mammal series will have a marsupial episode, and everything in between. The third reason that this episode is a new beginning of sorts is because this time I'm going to go even farther back in evolutionary history, long before the origin of the deuterostomes and the protostomes. In our taxonomy episodes, we just looked at these bilaterally symmetrical animals. Our exploration of animal taxonomy picked up at the end of the Ediacaran period, shortly before the Cambrian explosion. But there's a lot of animal life that was already around at the time. There's a lot of animals that are not protostomes or deuterostomes, because their lineages predate them both. Of course, I'm talking about some of the most primitive animals that still live on our planet today. The hydra, the jellyfish, the comb jellies, the sponges. In hindsight, I kind of regret not fitting them in earlier, although there are reasons why I didn't. We had just come off our multi-part series on animal physiology, which ended with an episode on the divergence and development of the deuterostomes and the protostomes. This was too good of a transition into protostome taxonomy to pass up, and I also had a hard time justifying spending an entire season on the taxonomy of these primitive animals. The sponges, for example, are pretty diverse, with a number of described species comparable to amphibians, around 8,000 or so, but the range is quite wide. The Cnidaria, like the jellyfish and anemones, have closer to 10,000 described species, on the same level of biodiversity as the reptiles or the birds. So in that sense, both the sponges of Porifera and the jellyfish and anemones of Cnidaria could theoretically have had a decent taxonomy episode of their own. But if we look at the ctenophores, there's a mere 125 or so known species. And there's a fifth as many known species of Placozoan, so a video on their taxonomy would be 5 minutes, tops, if I was really dragging it out. Now this is probably a case of me just tangling myself up in my own arbitrary rules and designs for this podcast, but however it happened, here we are, at our new beginning. And it's a perfect chance to introduce these most ancient animals and explore both their taxonomic relationships and their anatomy, lifestyle, and ecology. So, with all of this housekeeping finally out of the way, let's begin. Let's dive into the series on the Parazoans and the Metazoans, and the dawn of the lineage of complex animals that would come to dominate the planet. Our story begins 13,000 million years ago, in the final stages of the 2 billion year long Proterozoic eon. The Earth at this time was much different than it is today. There was much more surface area covered by water, as the crust was still forming. Proterozoic saw numerous pulses of where crust production surged and then tapered off, and then later surged again. This means that, by some estimates, more than 40% of the crust that exists today was created in the Proterozoic. This time would have been warmer on average too, except for the occasional ice age, which would become much more frequent near the end of the period, and have really important implications for the evolution of life, but we'll get into that later. We'll get into that when we come to it. Life had existed all throughout the Proterozoic, although for most of this time, up until about 1.3 billion years ago, that life was limited to simple single-celled microbes like bacteria and archaea. Methanogens and other anaerobic microbes were the first predominant type of life form on Earth, but they were usurped by oxygenic cyanobacteria a long time ago. Photosynthesis was also old news, as it had been around for about 2 billion years already. The oxygen crisis had come and gone, and then eukaryotes had emerged. And although we don't know for sure, a leading idea is that a symbiotic interaction over millions of years between a virus, bacteria, and archaea ended up shuffling genetic material around and integrating the bacteria into the archaeal cell. However it happened, though, the outcome was a larger, more complex cell with a highly organized internal environment. All of this was made possible by the symbiosis of the bacteria that evolved into the mitochondria, as it specialized in producing enormous amounts of chemical energy for its symbiotic host, the larger archaeal cell that it was embedded within. The increase in organization and complexity in the eukaryotic cells, as well as the surplus chemical energy, made these eukaryotes really adaptive, really evolutionarily plastic, compared to the bacteria and the archaea. The eukaryotes started to develop really unique traits and features that the world had never seen before, like the flagella. For most flagellated cells, the spinning flagella is like a propeller on the front of a plane, pulling it forward through the water. But in later groups, like the choanoflagellates, it became like the propeller on a boat that pushed it forward through the water. This simple adaptation of a flagella was revolutionary, as it immediately improved the cell's ability to capture food and move to preferable environments. In fact, it was so useful and so cool that a few million years later, one lineage would eventually double up and evolve 2 flagella, earning them the name Biconta. This Biconta lineage has flourished into a diversity of mostly single-celled eukaryotic organisms, but it's suspected that they may also be the ancestors of the Archaeplastida, which includes the green and red algae, and of course, all of the plants. Another massive breakthrough was the evolution of sexual replication. Before this, the planet had only known life forms that reproduced asexually, with cells dividing themselves to produce cloned daughter cells. But with the advent of sexual replication, 2 individuals could contribute a portion of their genetic material into a genetically unique offspring. This ability to replicate sexually and exchange genetic material to create new combinations, this made the eukaryotes much more genetically diverse, and thus much more resilient to pathogens like viruses, and it improved their adaptability and evolutionary plasticity in the face of changing ecological pressures. And of course, most importantly, the eukaryotes had evolved multicellularity, and not just once, but at least 25 times in different lineages. There are several hypotheses about how this happened. One idea was inspired from the dynamics of bacterial mats. As bacteria group together in colonies, their single-celled bodies and all of their secretions and released chemicals form a thick sludge-like layer over the ground. Within this film of slime, the cells eventually arrange themselves in layers. Those at the bottom are touching the substrate, like a rock on the beach, or a rock on the seafloor, or the seafloor itself, or wherever the bacterial mat is. These lowest bacteria may dissolve the mineral substrate through some sort of chemolytic or other metabolic process, and consume particles of food. Their waste products, and the debris from their chemical dissolution of the substrate, will diffuse upwards, where it feeds the next layer of bacteria, and their waste and debris feeds the next layer, and so on, such that a bacterial mat can eventually form a complex community with an organized flow of nutrients and energy between semi-discrete populations of different species or different genera of bacteria. This environment would create evolutionary pressures for the cells to adapt to a particular layer or a position in the mat, or some ecological role in this chemical food web. The cells would differentiate, to specialize and optimize their performance, and eventually they'd produce a group of cells that have to work together to sustain the environment they live in, or they all die. They'd be locked onto an evolutionary trajectory of cooperation that eventually had them merge into a coherent functional unit with differentiated cell types, the first multicellular animal. There's a few organisms alive today that do this, like some amoeba and slime molds. Two clades of amoeba are known to have cells that live individually, but they may emit pheromone signals telling other amoeba to pile on and conglomerate into a squirming slug-like mass called a grex. This larger pseudo-multicellular organism then hunts for food as an aggregate, as a kind of composite organism, so to speak, and diffusion helps ensure that every participating amoeba gets fed. The slime molds are usually single-celled, but they can enter a multicellular fruiting body stage in their life cycle, and when this stage ends, they revert back to independent single cells. So this is an interesting idea, that you have this colony of independent single cells, and as they work together and they evolve and adapt to complement each other in this microbial community, they eventually become a unified single organism. It's a neat idea, but this symbiosis hypothesis, if they're different species of microbe, or a colonial hypothesis, if they're the same species, this whole idea behind the origin of multicellularity, it has a lot of problems, a lot of flaws. The biggest issue with the idea is the consolidation of the genome. How did the genomes of multiple independent cells all get integrated together into a single coherent genome that's replicated and shared among all of the daughter cells? This is a hugely challenging question for the symbiosis hypothesis, like an archway trying to stand after the keystone has crumbled to dust. It forms a chasm of the unknown, separating two knowns. The two knowns are the existence of individual cells that can cooperate in a physically integrated group, and the other known is the existence of multicellular organisms. The unknown is the bridge between them. How did one develop into the other? Without evidence to shine light into this dark chasm of the unknown, the entire central premise of the symbiosis or colonial hypothesis remains a matter of complete speculation. Other ideas seek to address this issue, such as the syncytial hypothesis. This suggests that a single-celled organism produced multiple nuclei, which is a known thing that happens a lot, from slime molds to ciliates and other protists. And then the idea is it wrapped its multiple nuclei in their own membranes, perhaps as an evolved means of extra defense and regulation of what gets through to the genetic material, like regulatory molecules or environmental pollutants or viral proteins or whatever. These freshly individually wrapped nuclei were thus all part of the same mother genome, all part of the same original cell, but could eventually be adapted into a specialized cell of their own, thus creating a multicellular organism. However, there isn't a known example of this happening, and the supporting evidence, like the ciliates, aren't necessarily great support for this idea because their multiple nuclei are too specialized for them both to maintain their own cell. Like, a nuclei needs to have enough stuff to support itself, its own membrane and its own ribosomes and other machinery involved in expressing genes into proteins. But in these ciliates, some of their nuclei, they have multiple nuclei, but some of them are so specialized that they can't do that. They can't support their own membranes and their own functions as their own independent cell. They're so specialized that they just contain the genes for a particular function that the ciliate needs to use them for. So in a very similar way, this syncytial hypothesis also runs into a pretty deep issue. Another idea, one that I think is most plausible, is that a single individual cell experienced a mutation or a problem at some point in its replication cycle. The cell was undergoing division but failed to fully separate. If the cause of this failure was genetic in origin, then it's plausible that subsequent replication events, or attempts at replication, would create similar errors, and you'd end up with an embryo-like mass of replicating cells. For example, consider a gene involved in the creation and function of guanylate kinase. This is a molecule that helps recycle energy carrier molecules like ATP and GMP into their tapped products, ADP and GDP. A mutation to this gene could perhaps improve the efficiency of energy recycling and allow a single cell to support a larger multinucleated body that could theoretically be partitioned into a functional group of smaller cells. This idea is made even more compelling by the influence of predation. At the microscopic scale, single-celled predators hunting down single-celled prey can induce an evolutionary pressure in the prey to grow larger. We see this in vertebrate animals all the time, from the dinosaurs to elephants and whales, among many others. If the prey animal is simply too big for the predator to do any damage to it or kill it, then that in and of itself is a viable defense. And at the cellular scale, this phenomenon manifests as single cells like algae having been experimentally shown to develop dense little quasi-multicellular forms in response to threats from predators. Perhaps the most compelling evidence comes to us from viruses. Viruses infect larger cells, integrate their viral genes into the host's genome, and then uses the host's enzymes to create more of its own viral genes and viral proteins. A lingering after effect of viral infections is the presence of the viral genes in the host's genome. Apparently, 8% of our human genome is the accumulated contribution of viruses over the millions of years of our mammalian and primate evolution. Things get really crazy when you find out what these viral genes code for, and how these genes are used in the bodies of plants and animals today. Some of these genes have been modified and adapted for useful functions, while others were hardly adapted at all, because the function they served for the virus was valuable enough to preserve unaltered in the host. For example, viruses need proteins on their membranes that allow them to fuse with their host cell, and the genes for some of these fusion proteins have been integrated into the eukaryotic genome. The idea here is that viruses may have pioneered the evolution of fusion proteins, which were then used by the host eukaryote to create multiple cells that were basically all glued or stapled together. There's surprisingly strong evidence for this idea. Every known cell fusion protein in all multicellular life forms, from protists and plants to fungi and animals, are all derived from ancient viral genes. Think of something as ancient as sexual replication, where sperm fuses with egg to create a fused membrane and a new organism. That fusion involves proteins that originated with viruses. Because multicellularity emerged multiple times in different lineages, it's entirely possible that they didn't all do it the same way. Some lineages may have been spurred on to multicellularity only by predation, while others may have had a failure of separation during cellular replication, while others still may have some combination of multiple factors, and all of them were using viral fusion proteins in some capacity. As multicellular organisms, they would have enjoyed greater safety from predators, more efficient nutrient distribution through their bodies, and longer collective lifespans, as the aggregated whole, the mass of multiple cells, could live on even as individual cells within the collective die. In any case, by 1.3 billion years ago, life had evolved considerable complexity from its humble single-celled beginnings. No longer was the world just bacteria and archaea and single-celled, relatively simple eukaryotes, because now there was flagella-equipped sexually reproducing eukaryotes, and even multicellular organisms, which were becoming increasingly widespread and diverse. The reason I wanted to begin our story at about 1.3 billion years ago is because this is when a particular lineage of microbial multicellular life emerged, known as the Opisthokonta. These are the cousins of the amoebozoa. Now within this Opisthokonta group, there was a major divergence event that would create two massive clades that would come to dominate much of the Earth's biosphere. The Holomycota, which would produce the fungi and all of its relatives, and the Holozoa, which would create the animals and all of their relatives. Today's topic will have us following the evolution of the early Holozoa to explore how the first animals emerged from this curious lineage of flagellated amoeba cousins. Now at this point, we're still talking about relatively simple multicellular creatures. Some of the most primitive holozoans are the Mesomycetozoa, which are literally just clumps of spherical cells, like a bunch of grapes. These are known to parasitize fish by growing in their gills, and a few other animals as well. So I'm not sure what they were doing before fish were around. Probably parasitizing aquatic fungi, but who knows? These Mesomycetozoa are sister to a group called the Phylozoa, which includes all of the animals and their closest unicellular relatives, most notably the choanoflagellates. These guys appeared about 1 billion years ago, and they're a great example of the flagella-having, sexually reproducing eukaryotes. Well, they can reproduce sexually and asexually, but that just makes them an even better example of the evolutionary transition from asexual microbes to animals. Briefly, the choanoflagellates are oval-shaped cells with a flagella protruding from one end, and wrapped around the base of the flagella is a thick collar of protein-rich microvilli. As the flagella rotates to propel the choanoflagellate forward, it also creates a current of water that flows through the collar and into the body. Any debris or bacteria or other food particles are swept up in this current and ingested. Relevant to multicellularity, the choanoflagellates are born single-celled and live all of their lives as single cells. But they might spend some time in a colony, where a group of choanoflagellates will cement themselves together into a globular mass, where they can all waggle their flagella to feed. In so doing, they cancel out each other's movement, so they don't really move out of any food-rich areas that they happen to find themselves in. It's like the perfect way for a group of them to just hang out in one spot and eat. Now this is where things get wildly interesting. The sponges are the most primitive type of animal still alive, and thus they're the most closely related living animals to the choanoflagellates. And if we look closer at the tissues and the cells that make up the body of the sponge, we see something incredible. The inner layer, the lining of the gut of the sponge, is called choanoderm, and it's composed of choanocytes. These are oval-shaped cells with a flagella, and at the base of the flagella is a thick collar of protein-rich microvilli. As the flagella spin, a current of water brings food particles into the cell. It's exactly how choanoflagellates work, except instead of being single cells that swim freely through the water column, these choanocytes are organized into sheets of tissue, and food particles pass through this sheet into the rest of the body to diffuse and feed the rest of the body's cells. It's clear to see that the earliest animals were conglomerations of ancestral choanoflagellate-like cells all working together. But what's not clear is how this arrangement was achieved. This is the problem with the symbiosis hypothesis I described earlier. Lost to the fog of evolutionary history are the answers to how this transition between choanoflagellates and multicellular animals happened. What did this common ancestor look like? Did the members of some primitive choanoflagellate colony find themselves locked onto that evolutionary trajectory towards interdependence and eventual multicellularity? Or did one choanoflagellate have trouble separating and reproducing? Did predation encourage them to group up? We don't know. But in any case, it's extremely fascinating, cell-based evidence of the close evolutionary relationship between choanoflagellates and the animals. However, we should acknowledge some of the very important differences between choanoflagellates and the sponge's choanocytes, including major differences in the cell's structural protein composition, and the mechanics of the interaction between the collar and the flagellum. To put a date on this divergence, the choanoflagellates emerged about 1 billion years ago, as far as we can tell. But there's a broad range of estimates for the emergence of the animal lineage, with estimates as young as 650 million years old, to an older estimate of a divergence that occurred almost immediately after the choanoflagellates, 1 billion years ago. So think like 900 million to 1 billion years ago as being like the oldest estimates for when the earliest animals first emerged. Now, if we're talking about animals that are just alive today, the sponges of the clade porifera are the most basal. Their bodies are extremely simple, with an inner and outer layer of tissue held together with a stiff, collagen-rich matrix of connective fibers. The choanoderm is the inner layer of tissue that we talked about. It's lined with those choanocytes. All of their flagella collectively wiggling in the water creates currents, so water flows through the porous body of the sponge, and food particles can be absorbed in what is basically a scaled-up version of the filter feeding seen in individual choanoflagellates. Aside from this, the sponges don't really have differentiated tissues. They don't have nervous systems. There are really interesting, diverse populations of free-moving cells, amoeboid-like cells, residing within the connective tissue matrix, and they can move through it. But aside from these free-moving cells and that single epidermal tissue type that absorbs food and then that seals in the outer layer of the body, that's about it. That's all they have. They don't really have any other more specialized tissues. But as the main stem group of animals continued to evolve, more differentiated tissue types were developed, and internal body organization was improved. For example, the sponge's body is generated from a single type or layer of germ cells. This is fine for the sponge because they're just not very complex animals. They don't have differentiated tissues beyond a few basics. Tissues that serve specific functions like nervous tissue or muscle tissue or specialized organs. They don't have that. But as the animals evolved to become more complex, so too did their development, did their embryological development. Although this might be a question of the tail wagging the dog. It could be that a lot of the evolutionary changes we see in animals are the result of those species that were able to survive and propagate populations for some time as a result of whatever mutations and idiosyncrasies were going on in their particular genome and their embryological development. Like the embryological development and the things that happened there were the driver of the mature body's morphology and the relative evolutionary and fitness success of the animal. One of the biggest changes here was the evolution of a second type or a second layer of germ cells in the embryo, which facilitated greater tissue differentiation in the mature body form. These two germ layers are the ectoderm, which forms much of the outer tissues like the epidermis, and the endoderm, which forms the internal tissues like those lining the gut or the digestive system. These animals that possessed two germ layers are known as the diploblasts, and they represent the most primitive eumetazoan animals. These two germ layers produced novel tissue types, like the neural tissue and rudimentary sensory structures from the ectoderm, and the gut tissue from the endoderm. In addition, embryonic development was enhanced with a gastrulation stage, where the embryo changes shape from a ball into a hollow globular shape with distinct layers of cells. This improved embryonic organization directly facilitated more complex and more organized mature bodies. New ways to connect cells together were also developed, as were basement membranes that served as a novel substrate for tissue growth. That strange jelly mixture in the middle between the two outer epidermal sheets of tissue was gradually depopulated of cells, as the tissues themselves became more differentiated, and the extracellular matrix itself was being modulated with different types and concentrations of various connective fibers. More generally, the slow accumulation of mutations were expanding the genome of these early animals, equipping them with a broader suite of proteins, engaging in more complex interactions. For example, the sponges have many of the genes for proteins involved in the neural system of other animals, but they themselves do not have a neural system. This suggests that the sponges use these genes for some other purpose, perhaps as pumps and ion channels for maintaining internal pH and osmolarity, and these were simply repurposed for use in neural tissue in more derived animal lineages, like to maintain ion gradients that allow for action potentials. There's another really important genetic development that happened after the sponges, but I'll get to that in just a minute. The earliest diploblasts, or the animals with these two germ layers in their embryo, are the ctenophores, or the comb jellies, which are beautifully weird organisms that move ponderously through the world's oceans. Like the sponges, the comb jellies only have two layers of tissue, an inner and an outer layer, with the space between filled with a jelly-like matrix of connective tissues. Instead of choanocytes with flagella lining the internal body cavities, as is the case in sponges, the ctenophores have an inner body surface lined with cilia. Cilia are kind of like flagella, except they're smaller and much more numerous, like little hairs that wave back and forth to create currents. The ctenophores use the movement of these cilia to propel themselves through the water, mouth first, and to produce a current that brings food particles into their body. Ctenophores are super weird predators. They're entirely soft-bodied, and they have no teeth or claws or anything like that, as they feed on the small animals like rotifers and crustacean larvae that can get sucked up in their cilia-induced current. Some of them even have small retractable tentacles equipped with sticky pads for seizing larger prey. Food is brought into the mouth, where it's then ingested and dissolved in the stomach, and nutrients are distributed throughout the body via a series of short, broad channels, more akin to tube-like cavities than blood vessels. It's not entirely clear how the ctenophores evolved in relation to the sponges, although there's a few hypotheses that are worth mentioning. There's some controversy about the placement of sponges and ctenophores, as many molecular studies, particularly those that use more outgroups for contrast, tend to find that the ctenophores are the sister group to all the other animals, not the sponges. Again, this is somewhat controversial, and I don't think the matter is settled. This means that there's still a lot of mystery and ambiguity behind their evolutionary origin. Let's explore some of the ideas behind ctenophore evolution. Now, if the sponges came first, then it's possible that the ctenophores are a modified form of an ancient free-living ancestral sponge. After all, not all sponges are bound to the substrate. Some of them are free-swimming. Furthermore, some of the immobile substrate-bound sponges can reproduce through fragmentation, where a bit of their body breaks off but stays alive to implant somewhere else and then grow into a new sponge, presumably an immobile sessile sponge. Or they can reproduce through sexual replication, where a newly hatched larva is released into the water column to search for food before sinking and eventually binding to the substrate. In all of these situations, there's an ancestral sponge that's free-floating in the water. The ctenophores could plausibly have originated from a free-floating sponge, or from a sponge larva or fragment that simply never settled down on the substrate. Like, perhaps there was a community of sessile sponges whose larvae were evolving to be increasingly self-sufficient, and while some of the larvae kept settling down to produce more adult sessile sponges, some of them diverged to keep swimming. They gradually lost the filter-feeding features of the sessile sponges, and they developed traits better suited to more mobile, free-swimming predatory organisms. It's also possible that they kept this sessile adult sponge form, but that it was just pushed back to be a very small, terminal portion of their life cycle, and the vast majority of their life was spent in the larval stage. We see a similar life cycle phenomenon in various other animal groups, like various arthropods, like the dragonflies. Now, on the other hand, if the ctenophores came first and the sponges diverged after them, then perhaps some ancestral ctenophore found that it could get by just fine if it conserved its energy by staying in one place and letting its food come to it, and this tendency developed into a fully sessile filter-feeding lifestyle. Extra features that were no longer needed for swimming or hunting were lost, and they became much reduced in form to become the sponges that we know today. Although there's other evidence, like various features of the cellular physiology and details of embryonic development, that suggest that sponges are indeed the older lineage, and the ctenophores diverged later and are, in fact, more closely related to the cnidarians, or the jellyfish and the anemones and all of the other animals. In any case, after the ctenophores had established themselves, the stem group diploblasts continued to evolve, and they developed a number of important gene families. Perhaps the most well-known of these are the Hox and Parahox genes, which play a critical role in the arrangement and proper development of the body, particularly its repetitive or segmented parts, like the bones and vertebra and the ribs, and the shape, size, and placement of various appendages. The Parahox genes, for example, may have originally been used to organize the development of the digestive system, from the mouth to the stomach, and then when a bilateral body form was evolved, they regulated the development of the digestive tract from mouth to anus. These Hox and Parahox genes are extremely important to all bilaterians, the bilaterally symmetrical animals, and they have their origin here in the ancestral cnidarians. The cnidarian clade includes the jellyfish and the sea anemones, and much like the comb jellies, they are radially symmetrical. They don't have a front or a back, or a left or a right side. They have a top and a bottom, but like a plate or a bowl, they look the same no matter how you rotate them. Where the ctenophores had a specialized cell type that was like a sticky pad for capturing prey, the cnidarians have a specialized cell type called a cnidocyte. It's a weaponized cell that possesses a spike or a spear-like tubule that can be explosively ejected, inflicting a painful sting on the target organism. The sting comes from the fact that these cells are venomous, and they can injure or kill the prey with various neurotoxins. Also like the comb jellies, the body of the cnidarian is composed of two types of tissue, like an inner and outer lining, with a non-living matrix of connective tissue sandwiched between them, to give them some volume and some structural integrity. A more unique and characteristic trait of cnidarians are their many tentacles armed with their stinging cnidocytes. In the jellyfish, these tentacles descend down from the main body, while in the anemones, the body is anchored to the ground and the tentacles reach up to wave in the water above it. These tentacles can sting and stun various prey animals, rendering them helpless as they get dragged into the cnidarian's mouth. Earlier, I described the cnidarians as radially symmetrical, and they are as adults. But there's some nuance here. In the larval stage, their body form is bilaterally symmetrical. It looks like a small, slug-type creature, with a head and a tail, and left and right sides. Where the adult cnidarian has nervous tissue arranged in a diffuse nerve net, distributed more or less equally throughout the body, the larval cnidarian has a much more centralized nervous system structure, with a distinct mass of neural tissue near the head. This cephalic ganglion in the cnidarian larva is the first attempt at a centralized nervous system in the entire lineage of Metazoa. It's the first attempt by evolution to create an animal brain. Now, this is an incredible fact all on its own, but it gets crazier. This developmental pattern suggests that bilateral symmetry was evolved earlier than thought, perhaps in the common ancestor of cnidarians and the rest of the animals. The cnidarians thus represent an intermediate position in this evolutionary transformation, where bilateral symmetry had been achieved for the larva, but was not maintained through metamorphosis into adulthood. Only in the bilaterians was this bilateral symmetry sustained from birth to maturity. Also, recall that the ctenophores and cnidarians are diploblasts. Their embryos possess two distinct germ layers, or groups of stem cells that differentiate into the body's tissues. The bilaterians are triploblasts, which means their embryos have 3 germ layers. This new third layer that they evolved is called the mesoderm, and it forms intermediate tissues in between the epidermis and the gut. So, like all of the bones and the muscles and the organs and whatnot. Well, it turns out that many of the genes and the regulatory programs involved in building the mesoderm were already present in the cnidarians. This is to say that the jellyfish are not themselves triploblasts, but they possess many of the genetic resources necessary to make mesoderm germ cells. This is a legacy of their shared common ancestry with the triploblast bilaterians. If you recall the Hox and Parahox toolkit genes I mentioned earlier, these are also present in the cnidarians, further reinforcing their close phylogenetic relationship with the bilaterians. Okay, let's step back for a minute. All of this evolutionary change that we've talked about today, from the development of photosynthesis, multicellularity, and sexual reproduction, ultimately led to the emergence of the Opisthokonts, and after the ancestral fungi diverged, from the stem group evolved the Metazoa, the animals. The earliest animals, so early and primitive that they don't even qualify as true Metazoans, belong to the ancestors of the sponges and the sponges themselves, those mostly sessile filter-feeding pillars of simple flesh. The true Metazoans emerged when their ancestors detached from the substrate and swam freely in the oceans, forming the ctenophores, the comb jellies. Accumulating genetic complexity produced the cnidarians with their Hox genes and blueprints for the mesoderm, and sister to them are the true bilaterians. All of these primitive animals filled the world's oceans in a time known as the Ediacaran, which lasted for about 100 million years, from 635 to about 539 million years ago, roughly. This was a strange and fascinating moment in Earth's history. Life had achieved a level of complexity that produced all manner of weird macroscopic forms, from disc or bag-like animals, slug-like creatures burrowing through the sediment, to strange and cryptic organisms with a body form that's been described as quilted or mattress-like. All of these strange creatures flourished in this time immediately before the Cambrian explosion. This was a major diversification event that spanned many millions of years, where the ancestors of all modern animal phyla emerged. The Cambrian explosion would mark the end of the Ediacaran, and the end of most of the Ediacaran life forms, who were outcompeted and driven to extinction by the new types of animals on the scene, like the vertebrate fish. But some Ediacaran creatures survived, their lineages perpetuating into the modern day, with much fresh biodiversity generated in the eons after the Cambrian explosion. These are the sponges, the ctenophores, the cnidarians, and all of their close relatives. This has been the story of the early evolution of the Metazoans, and in the following episodes we'll dig deeper into each of these groups. In the next show, episode 98, we'll explore the physiology, life cycle, and ecology of the sponges. Episode 99 will explore the ctenophores, and then we'll have a big surprise for episode 100. After this series is over, we'll move back into the realm of the protostomes and deuterostomes by exploring the physiology, life cycle, and ecology of the mollusks and crustaceans, and the insects, and the fish, and the reptiles, and everything else. If you want to check out these episodes right when they're posted, then subscribe to the channel. If you liked this episode, then hit the like button or give it a 5-star review. All of this stuff really helps the podcast get boosted up in the algorithms, so more people can find it and enjoy their own audio adventure into the world of the biological. Check out the official store, tell your friends about the show, and if you're super awesome, then you'll become a Patreon supporter. There's tiers as low as $1 and $2 a month, so if you think that's worth it for a monthly biology episode, then sign up. It means a lot to me, and it really helps me keep the lights on and cover the monthly website hosting fees. And as always, thank you for listening.